Today we're going to be talking about a monohybrid cross. And a monohybrid cross is a cross or mating between two organisms that differ in only one character. For example, height. Now, back in the mid-1800s, Gregor Mendel did some work with pea plants. And we're going to repeat some of the experiments that he actually did. One of the experiments he did was he took a tall pea plant and crossed it or mated it with a dwarf pea plant to see what the effects of that cross would be. So we're going to go ahead and we're going to start out with that particular experiment. Now I'm going to start by writing down a P, a capital P, that represents the parent generation. And then we're going to go ahead and we're going to write phenotype. So what are the phenotypes of these particular pea plants? So a phenotype is what that organism actually looks like. So we're going to go ahead and we're going to take a tall pea plant and we're going to cross it or mate it with a dwarf pea plant. Now I'm going to go ahead and write down genotype. These are the genes that these plants actually contained. Now Mendel didn't know about genes. He referred to these genes as factors back then. What genes does the tall pea plant contain? And I only care about, the genes for tall here. Now before we can write down genotype, we have to pick a letter that represents that gene. Now remember, a gene is just a sequence of nucleotides that codes for a protein. In that case, this protein is going to make this pea plant tall. So I want to use either a T or a D to make things easy for myself. So which one do I pick? I'm going to go ahead and I'm going to pick the D. And the reason is because not only am I going to use the letter D, but I'm going to use a capital and a lowercase. And it turns out a capital D and a lowercase d look a lot different than a capital T and a lowercase t. So I'm going to go ahead and I'm going to pick D. Now, the capital D is going to represent a dominant gene. Now Mendel also figured out that there could be two different types of genes here. In this case there could be a tall gene and a dwarf gene. It turns out that tall is going to be dominant to dwarf. That means when the two genes are together, the tall gene masks the effects of the dwarf gene. So all you get is tall pea plants. Now again, Mendel had to figure all this out through experimentation. Let's go ahead and write capital D, capital D. Now another thing that Mendel figured out, and this is why he's referred to as the father of genetics, he figured out that it took two genes, so two capital letter D's here, to code for a trait, or this is referred to as a trait. Tall is a trait and dwarf is a trait. These are traits of the character height. Now, dwarf is recessive, so I'm going to use two lowercase d's to represent a dwarf plant. And now I'm going to go ahead and I'm going to write gametes. Gametes are sperm or egg, and yes, most plants produce sperm and egg, especially pea plants. Now this tall pea plant could either give a gamete a capital D or it could give this capital D. Remember, you can only give what you have. So this tall pea plant only has capital D's, or genes for tallness. Now the same thing goes for the dwarf. When this gamete is produced, it could contain either a lowercase d or a lowercase d. Now remember, the gamete is going to contain lots of different genes for different characters. But we're only talking about the height character here. So in terms of height, this dwarf pea plant could contribute a lowercase d or a lowercase d. Now we're going to make a Punnett square. And a Punnett square was developed later on after Mendel did his work by a guy named Reginald Punnett. And a Punnett square is going to be used to determine what combinations of offspring could be produced from the different gametes. So I'm going to go ahead and I'm going to draw a Punnett square. Now this Punnett square is going to be for a monohybrid cross, so it's going to only contain 4 squares. So I'm going to take these two gametes, or these two genes, and I could either put them here and here, or I could put them here and here. Doesn't matter. The results are going to be the same. So I'm going to take my D's, and I'm going to put one of the D's here, and the other D down here. And I'm going to take my lowercase d's, and I'm going to put one here and one over here. Now, what I do next is I just figure out, what if this gamete containing a capital D got together with this gamete containing a lowercase d? What would the offspring, what would be the genotype of the offspring? And it would be capital D, lowercase d. And the same thing goes here. What if this gamete containing a capital D got together with this gamete containing a lowercase d? Again, the genotype of that offspring would be capital D, lowercase. And the same thing goes here. Capital D, lowercase. And the same thing goes here. Capital D, lowercase. Now I want to go ahead and I want to summarize my results. So these genotypes represent the offspring of the parents. So I'm going to go ahead and I'm going to refer to them as the F1 generation. The first filial, meaning the first generation, the kids of the parents. And these are genotypes, so I'm going to go ahead and I'm going to write genotype here. Now, it turns out that 100%, or 4 out of 4, are going to be capital D, lowercase. That means they're going to be heterozygous. They've got one of each type of gene here. So I'm going to go ahead and I'm going to write 100% are going to be capital D, lowercase. Heterozygous or hybrid. Now I'm going to go ahead and I'm going to write phenotype. So based on these genotypes, what would the phenotype be? Now we know, like I told you, tall is going to be dominant to dwarf. So that means we've got two different genes, so only the dominant gene is going to be expressed here. The dominant gene is going to code for tall. So 100% of our plants are going to end up being tall. And again, Mendel discovered this through experimentation. Now, this is where things begin to get interesting. Mendel didn't stop there. He then took two of these kids, and it doesn't matter what two kids you take, because they all have the same phenotype, they all have the same genotype. So we're going to take any two. And again, it doesn't mean that we just have 4 offspring. These are the various combinations. In this case, they all happen to be the same. I could have had 100 kids, I could have had 50 kids, I could have produced 1,000 kids, 1,000 different peas or seeds. But again, it doesn't matter. All I'm going to do is I'm going to take 2 of these offspring, however many that were produced, I'm going to take 2 of them and I'm going to mate them together. So I'm going to come over here and I'm going to go through the same format that I have here. So I'm going to refer to these as the F1, because they are the F1s. And I'm going to go ahead and I'm going to switch things up a little bit. I'm going to write phenotype first. You don't have to, but that's my preference. Phenotype. And I'm going to cross a tall with a tall pea plant. Now their genotypes, again, are all going to be the same. They're all going to be heterozygous. So I'm going to write genotype and capital D, lowercase. Cross that with a capital D, lowercase d. And now I got to go ahead and I got to write gametes. So what are the gametes that these two plants could produce? Well, this tall pea plant could end up giving its gametes a capital D, or it could give its gametes a lowercase d. Same thing goes for this tall pea plant. It could give a capital D to a gamete, or it could give a lowercase d to a gamete. Now I'm going to go ahead and I'm going to draw another Punnett square. I'm going to go ahead and take these gametes, capital D and lowercase d, and now these, capital D, lowercase d. And I'm going to go ahead and ask, what if this gamete containing a capital D got together with this gamete containing a capital D? What would be the genotype of the offspring? Capital D, capital D. Now I do the same thing here. Capital D, lowercase. Now, When I get here, again, by convention, that means the scientists decided it should be that way, the capital D gets written first. So capital D, lowercase, and then here I got two lowercase d's. Now I want to go ahead and I want to summarize my results. So these are genotypes. They're going to be the genotypes of the F2 generation. In a sense, this is the second filial generation, the grandkids of the parent generation. So again, these are genotypes. So I'm going to go ahead and I'm going to write genotype. And it turns out that I have three different genotypes here. 1/4 of my offspring would be expected to be capital D, capital D. So 25%, or I could write 1/4, doesn't matter, are going to be expected to be capital D, capital D. And it turns out 50% are going to be expected to be capital D lowercase, or heterozygous. 50% capital D lowercase. And then 25% would be expected to be lowercase d, lowercase d. Now, when you've got two of the same letter, that's referred to as homozygous or pure breeding. So these are referred to as homozygous recessive, where this would be homozygous dominant. So those are my genotypes. Now I want to go ahead and, what phenotypes would I get from these genotypes? So I'm going to go ahead and I'm going to write phenotype. Now it turns out that a capital D, that's going to give me a tall pea plant. And also capital D lowercase, because the capital D is dominant to the lowercase d. The tallest gene is dominant to the dwarf gene. So again, this genotype right here would also give me a tall pea plant. So it turns out that 75% of my pea plants would be expected to be tall. And 25% would be expected to be dwarf. Now again, in order to have a dwarf pea plant, I have to have two recessive genes. Because if I so much as have one dominant gene, it's going to mask the effect. So it takes two recessive genes to get a recessive phenotype here, a dwarf. Now, I could take this a step further and I could write phenotypic ratio. So let's go ahead and do that. Phenotypic ratio. Phenotypic ratio. So what's the phenotypic ratio? A ratio is a comparison of one thing to another. So we're going to compare the different phenotypes here. So what I'm going to do is I'm going to take the 25%, the smallest number, and I'm going to divide it into the largest, and then I'm going to divide it into itself. So 25% goes into 75% 3 times, colon. And then 25% goes into itself once. So my phenotypic ratio is 3 to 1. That means I've got 3 tall plants for every 1 dwarf plant. That's a phenotypic ratio. And I could also do it with genotype if I wanted to. The genotypic ratio here would be what? 25% goes into 25% once, 25% goes into 50% twice, and it goes into itself once. So it'd be 1 to 2 to 1. That would be the genotypic ratio here if I wanted to do it. Finally, I could take this one more step further. And I could say, what if I produced 100 offspring? What if 100 F2 offspring were produced? Okay, again, this doesn't mean that I produced 4 offspring here. These are just the different combinations that could be produced. Let's assume that I produced 100 F2 offspring, 100 seeds. When I plant those seeds, how many of those seeds would be expected to be tall? Out of the 100, how many would be expected to be tall? Well, what I'm going to do is what's 75% of 100? 75. I would expect 75 of those seeds to result in tall pea plants. And 25% of those seeds, or 25 out of the 100, to result in a dwarf pea plant. Now again, I could do this with a genotype. So if I genetically analyze these seeds, these peas, How many out of those 100 would be expected to be homozygous dominant? Well, 25%. So 25% of 100 is 25 Ps. So 25 Ps would be expected to have this genotype. 50% would be expected to be heterozygous, and 25% of those 100 Ps would be expected to be homozygous recessive. And that's going to be it for monohybrid crosses.